So pollination by water in valleys area you see in the first diagram. Then second is insect pollination. Now in the first diagram it is male flower and there is stigma and male flower, then female. So outbreeding devices you see, then pollen pistil pollination, pollen grain germinating on the stigma, then pollen tube growing through the style, then pistil showing path of pollen tube growth in the third diagram, an enlarged view of an egg apparatus showing entry of pollen tube into synergist. This is the D diagram. And in the E diagram, discharge of male gametes into a synergid and the movements of the sperm, one into the egg and the other into the central cell. So A second diagram, then C third diagram, you see stigma, then style, then ovary. So you see pollen tube, then antipodal, polar nuclei, egg cell, synergid. So longitudinal section of a flower showing growth of pollen tube. So central cell, egg, then two synergids, then plasma membrane, egg nucleus, filiform apparatus guiding the pollen tube, male gametes, and then vegetative nucleus. Now there is artificial hybridization, includes emasculation, bagging, dusting and rebagging, then double fertilization, includes syngamy and zygote, primary endosperm nucleus, triple fusion, double fertilization, primary endosperm cell PEC, endosperm, zygote into embryo. Fertilized embryo sac showing zygote and primary endosperm nucleus PEN. Stages in embryo development in a dicot. So zygote, globular embryo, then heart-shaped embryo, and then last one, that is radical and plumule having cotyledons. It is mature one having suspensor cells. And in the first diagram you see degenerating synergids and zygote 2n primary endosperm cell PEC having primary endosperm nucleus, that is triploid, and then degenerating antipodal cells. Now post-fertilization structures and events. So post-fertilization events includes endosperm, then embryo. So in embryo you see embryogeny, it is the study, and zygote shapes, proembryo, then globular heart shape, and mature embryo. Embryonal axis has two cotyledons, epicotyle having plumule, and hypocotyle, then radical or root tip covered by root cap. See in the diagram a typical dicot embryo you see having plumule, then cotyledon, hypocotyle below it, radical having root cap. In second diagram, LS of the embryo of grass, the scutellum, coleoptile having shoot apex, epiblast, radical, root cap, and coleorhiza and coleoptile. Seed are non-albuminous or albuminous. There is perisperm, dormancy, pericarp, it is fruit ball, then false fruit, true fruit,